Hello everyone, and welcome to another video. Today we're going to talk about morphogens and signaling, specifically when to use which terminology, because these are two things people mix up a lot. So we'll go over what the two are, and then a bit more specifically how morphogens work and what they're responsible for. So, signaling molecules. A signaling molecule is a molecule that triggers a change or some activity in a cell, induces some sort of response. So this can happen by the molecule, which in this drawing here is this red square or diamond, can either bind to a receptor and trigger some activity within the cell, or it can actually be taken in via endocytosis or pinocytosis, and then trigger some activity within the cell. Signaling molecules are prevalent everywhere. They are responsible for triggering gene expression, propagation of action potentials in neurons, so the neurotransmitters that you find in the synapse, those are signaling molecules. In immune responses, when an antigen binds an immune cell and triggers some activity, that is a signal, and then many other countless functions. So they're involved everywhere. Now, morphogens are actually a signal. They are signaling molecules, but a very specific kind. So you could say all morphogens are signals, but not all signals are morphogens. That's important to keep in mind. So a morphogen, what it's responsible for is defining cell fate, typically in embryonic development. So specific concentrations of these morphogens, and they'll often be proteins that act as transcription factors or something like that. Dependent on their concentration, they will define different cell fates and activate different genes. And what they'll do is actually form a gradient across an embryo, and then different regions will have different cell fates. So let's look at how this works with a very popular model called the French flag model. I will point out this isn't perfect, but it kind of gives a good starting point for understanding how the concentration dependence works. So we use a French flag because of the three regions of different color, which you can think of as three different cell fates. Then the graph underneath shows the different concentrations of whatever our morphogen is and how it activates these different fates. So in the most anterior, which we'll call the left, and posterior on the right, at the most anterior, you have a very high concentration of this morphogen. And most importantly, it's above this higher threshold. Now, if the concentration of this morphogen is ever above this threshold, it will activate a particular gene and cause this blue cell fate. Now, as you go further down, further posterior, the concentration is a bit lower. And in between these two thresholds, you'll have activation of gene 2, which will give this white cell fate. And as you get more posterior and the concentration is even lower, you get the concentration below all the thresholds, in which case it'll just be the default state. It's not activating any genes that it normally would be. So because of that, you get these three regions of distinct cell fates, just dependent on the concentration of a single morphogen. Now this can be in combinations, and usually there's more than one morphogen, and we'll go over an example in Drosophila in a moment. But in the simplified model here, it demonstrates how different concentrations give different fates. Now in Drosophila, It's an interesting case because in early development, when normally an embryo would undergo cell division from 1 cell stage, 2, 4, etc. The early Drosophila embryo undergoes division to a point where the nuclei divide, but the cell does not. So you end up with a syncytial cell, where it's a single cell that's multinucleated. So this Drosophila embryo here is one cell with many, many nuclei inside. And what this allows for is these morphogens. So the two in this example are bicoid and nanos. Bicoid represented by blue at the anterior and nanos by the red in the posterior. We do actually have an entire video devoted to anterior-posterior patterning in Drosophila, where we go over not only these two morphogens, but many others that are involved in this process. But for simplicity's sake, and to get the point across about concentration, we'll just look at these two. So in the early embryo, the mRNA for bicoid and nanos are deposited on either end. So the bicoid mRNA is deposited in the anterior, the nanos is deposited in the posterior. This RNA comes from the mother. And then what happens is when it's translated, it diffuses down the embryo. So that's why you get a higher concentration at the anterior, but then the bicoid protein diffuses down and slowly gets less and less concentrated. And same with the nanos. So because of this, you have varying concentrations from anterior to posterior and posterior to anterior, and this allows for the differential development of that axis. So the highest concentration of bicoid, as well as some other morphogens that again we're not going to talk about, you get anterior structures. Now the higher concentration of nanos and lower bicoid, as well as again some others, you get more posterior structure development. So this is how morphogens are responsible for differential cell fates and differential development dependent on their concentration. And Drosophila is kind of a perfect way to show that because of this unique syncytial cell. So the main takeaways from this are that morphogens are a signal, but they're a very specific type of signal. So if you're talking about development and the signaling molecule you look at is acting in a concentration-dependent manner, then that is likely a morphogen. If it is not concentration-dependent, then it is not a morphogen. It is some other type of signaling molecule. And it's important to be able to differentiate those. And they are responsible typically for early patterning in the embryo and axis development. Thank you for watching, and I'll see you next time.